Um, the next speaker today is Dr. Devaki Bhaya. She's a research scientist at the Carnegie Institute, and her research interests are on cyanobacteria. She's mainly interested in the genomic diversity of these organisms in different kinds of environments, in extreme environments, and under stress. And she's got a long-standing interest in the biochemical networks that govern phototaxis in these microbes. And she's the next speaker. Devaki.
So can you hear me? So it's a pleasure to be here, and thanks for the invitation to the organizers, and I'm hoping to learn a lot from this. I already have from the first talk, but I'm going to bring you back from technology nirvana to the beastly world of biology, which is difficult and but fascinating, and that's what I'm going to spend the next half an hour trying to tell you. Let's see, where's the mouse? So I've been working for the last 10 years as part of a big group looking at the microbial communities. Here we go. In Octopus Spring, and some of you may know that Octopus Spring was where Taq polymerase, which is the life of PCR, was actually discovered. But we look at the cyanobacteria in these hot springs, and all of this color you see around here is where bacteria start to grow. And we have focused on this very little region from about 70 degrees to 50 degrees, where cyanobacteria seem to really be able to do extremely well. And the other thing I want you to keep in mind as we look at these microbial communities is that there's a lot of fluctuation that's going on in terms of light, in terms of temperature, and then there are very predictable fluctuations, for example, the day-night cycle. So anybody who has to do well there has to be able to take all of this information and be able to coordinate it into some kind of response that allows them to survive in these very difficult circumstances. So what I want to introduce to you, because I don't think many of you think about cyanobacteria every day, is that they're a very ancient and interesting phylum. They're photosynthetic, and if for nothing else, you should remember the fact that if they didn't figure out how to do photosynthesis by splitting water and producing oxygen, we wouldn't be here, because that's the oxygen we breathe. They're also primary producers in many, many environments, including the terrestrial and the marine environment. And that's important too, because as the climate changes and warms, there's going to be impacts of the fact that the primary producers are going to change their efficiency. It also leads to things like toxic blooms, which I'm sure many of you have heard about. Apart from being able to fix carbon, they can also fix nitrogen. So they make really good partners in any kind of symbiotic relationship because they can produce and fix carbon as well as nitrogen. And if you've been around for 2 billion years, you get pretty good at being resilient. So they're cold tolerant, they're heat tolerant, and these are things we know very little about the molecular responses. As a consequence, in many countries, they're used as biofertilizers. And they're a rich source of very novel compounds because they do a lot of secondary metabolite production. Obviously, I wouldn't be here if you couldn't genetically manipulate them. And there's literally now hundreds of genomes that are available. So with that introduction, I'm going to skip right into telling you more about the stories I'm going to focus on today. And I only have half an hour, so I've got to compress a lot of information, but I hope you can come back and speak to me later on. And the first story is about the diversity in these environmental biofilms, and there's 2 studies that I want to focus on. And then I'm going to tell you a little bit at the very end about the roles of phages and the coevolution of populations, because phages are so important in these environments. And I'm fortunately not going to tell you very much about our studies on environmental stress and on group behavior, particularly phototaxis, but hopefully I'll get a chance someday to do that. So if you want to query microbial diversity, there's many, many ways to do it. And as Jean said, you can sequence now anything at great depth and detail. But it all started out really many, many years ago by just doing 16S rDNA and the genomes of model organisms You can then move on to something called the tree of life, which is to fill in all the phyla we know very little about. And as you know, there's many, many environments. For example, the Earth Microbiome Project is trying to look at microbiomes in very many different parts of the world. You can get a little more detailed, and you can start looking at sequences of related clades. And you can also look at populations within a particular species, within a species or within closely related communities, which is a metagenome. And it's also possible now, of course, to go to genomes of single cells. So this is just to give you a sense, which many of you probably know, that you can now, it's easy to do the sequencing across any dimension you feel like. What I think, and again, this is something working off what Jane just said, it's the analysis part that is really very tricky, and where I think us as biologists need to collaborate effectively with computational biologists as well as people doing bioinformatics. So here's where my story begins, and you don't have to pay attention to the names. All it shows you is that if you use the 16S rDNA phylogeny to look at cyanobacteria, you can see immediately that it's very deeply branched. And I want you to focus just on this little piece of the branch. All the OS stands for octopus spring. So even within the spring, there's a lot of diversity. The second thing to note is the one that I've pointed out in blue and the one in red. So in that channel that I was talking about in Octopus Spring, there are guys that you can always find at about 50 degrees, whom I'll call for the rest of this talk Synechococcus OSB or SYN-OSB. And then there's another ribotype that you often find at the high temperatures at about 70 degrees. And this was the start of what we really got interested in. Why can you always go back year after year and find the same ribotypes at very particular parts of this thermal gradient? So very simply put, very naive questions that we started out about 10 years ago is asking, do these different ribotypes that we can now establish really reflect functional diversity and specialization across the temperature gradient? Because those are just like very little pieces of information. We want to fill in that information. And more excitingly, rather than just get more and more sequence information, can we begin to start to look at the rules of community behavior at a molecular level? And I wanted to do this both in the context of biotic and abiotic stresses in the environment, because working with the model organism often doesn't allow you all of these other pieces of information that you can use. So one quick slide to summarize a lot of work that went into this. And how did we do it? You just go into the mat. You can go into various temperature parts of the mat. You get these things called core samples, which is about the size of my little fingernail. And you can grind that up, get DNA from it, which would be the metagenome. You can also do what are called enrichment cultures, where you try to get a particular type of organism, in our case, obviously the cyanobacteria. And as you can see here then, we got sequences from OSB and from OSA, the high temperature and the low temperature. We've also, in my lab, paid particular attention to the fact that if we really want to go further, we want to build an experimental system we can manipulate in the lab. And I'm happy to say we're actually able to do this. We are able to genetically manipulate these organisms, and that sets up a really nice experimental system where any hypotheses we make based on what's happening in the environment, we can actually try to check in lab conditions. So jumping right ahead, what happened when we actually did the genome sequencing of Synechococcus OSB and OSA? And I can show you here, there are only 3 megabase genomes. They're small. They have about 3,000 genomes. But what was really surprising about this, and this is shown, and I'm sure many of you are familiar with this, if you look at the outer ring, that's OSB. And if OSB and OSA have a very similar gene content, then and in terms of synteny, you'd expect these two, the outer and the inner circle, to look pretty much the same. But even if you're colorblind, you can actually see that that's not the case. It's almost as if the genome has been snipped up and then rearranged. So that's relative to each other. There's almost no synteny maintained between these two organisms, which was very, very surprising to us, and this was about 10 years ago. And however, we did see that there's a large number of transposons and repeat elements which might explain part of what's going on. And we also had evidence for extensive horizontal gene transfer and a very high level of diversity, which is what I want to explain to you a little bit more. How did we investigate this diversity? And this was long before any metagenomes. This was about 10 years ago. The first metagenomes were really being established. And we sort of went along and made the rules as we got our data. And all we had, it seems kind of pitiful now, we had only 200,000 paired and Sanger reads. But what I'll try to explain today is even with those reads, and they were pretty high quality, you can use them in various intelligent ways if you have particular questions you want to ask. So what we did is we took all of those paired end genome reads and we put it through a filter criterion, which is that we wanted them to match exactly either the OSA genome or the OSB genome. And we use very, very stringent criteria to be able to do this, and about 40% of those reads did fit in. But surprisingly to us, and maybe not so surprising to you now, because I've told you that these genomes are extremely scrambled, about 60% of those reads actually did not match either the anchor genome OSA or the anchor genome OSB. And we call this somewhat unfortunately the illegal clones because they didn't fit our criterion. Now, what do these illegal clones really tell us? And I'll give you an example of how we've been able to use this data to derive something about that whole population. So you can look here now, and you see here's the reference genome, for example. It has about 4 genes. We're just looking at a little piece of the genome here. And what you can see here, so let me go back. What you see here is that we had these so-called illegal clones, where we would find one end, for example, this blue arrow here, completely matching a part of the genome, and another read, say, for example, that completely matched the green arrow. But the other end of these paired end reads didn't match anything at all. So there was no way we could put it onto the anchor genome. And this was a whole very large bundle of reads that we never really knew what to do with until we were able to sequence some more longer library pieces. And then we found, aha, here's what's happening. In fact, this whole region, which is about 5 kb, has been inserted into this part of the DNA. So there's a cell out there that has picked up, thanks to a transposon, a piece of DNA that in fact has functionality. So what we were able to do then is to show that in fact these 2 genes, the FAO-A and the FAO-B, which are required for ferrous ion uptake, are actually being expressed in the mat itself. So we did what is called in situ transcriptomics, where you can see that these 2 genes are in fact going up around dusk and then fall off at night, potentially when there's more of a chance to be able to uptake ferrous ions. So what I want to leave you with the impression is that even though we didn't have this particular genome, we were able to use the metagenome or exploit the metagenome to find out a functionality that existed in the population, but not in our reference genomes. And there are many, many other examples of this. So now I want to shift gears to the second story. So to complete this story, what I'm really trying to tell you here is that a combination of using anchor genomes and reference genomes with metagenomic data, even if it's not very deep, can elicit information if you look very carefully and follow it up with experimental data. And now I'm going to go into something that's really a work in progress, and that's done in collaboration with Daniel Fisher, who's also part of this group. And it's trying now to look at microdiversity in the cyanobacteria, because we had hints from the metagenome data that not only were there these large-scale rearrangements, there was also diversity at individual loci. And what we tried to do there is we picked a lot of loci and we did deep sequencing, Illumina sequencing, which then we cleaned up with a program that Mike Rosen in Daniel Fisher's lab has figured out called DADA. And we ended up with so-called inferred alleles, which have single nucleotide polymorphisms. And what that immediately told us that apart from these 2 anchor genomes and metagenomes, we could also start looking with a great deal of detail at individual loci. And now what I'll try and tell you is what we actually found from something like that. So here are just two examples, although, of course, since we sequenced 100 loci, we have 100 such images. Here's the 16S rDNA, which I'm sure all of you are familiar with. Here's the 3% operational unit, which tells you what a species might look like. And comfortingly, you can see that here's OSB, the blue ring, and it's also the most abundant number of reads, which is what we'd expect because we know OSB is one of the most abundant guys in the mat. And you can see that OSA, which we expect is pretty far away, and there are about 40 other alleles. Now, this is looking at deep sequencing. You can take another loci. This is just a protein orf. We don't need to worry about details. And you can see here that now we have about 85 different alleles. Here's OSB for reference, but now it is no longer the most abundant allele. And here again, just for reference, now that you know these guys, here's OSA, the one that's very, very different. So for each of these different loci, we now have evidence of this kind of diversity at the nucleotide level. We're in the process now of figuring out what that means at the protein level, and all the loci we chose also have crystal structure, so we can layer that information onto the structure of the protein to say, is this really making a difference to the behavior of these organisms? And just to finish up this part of the story, which is still a work in progress, I wanted to show you another way we've tried to visualize this data. And I just want to throw out this idea that, in fact, one of the things that is something I'd love to get feedback on is that when you have so much data and you really want to show it in a concise form, it's extremely challenging because you lose pieces of information. But here, what I'm trying to show you, for example, is here's the Synechococcus genome position from 0 to 3 megabases, which is what it is. And here are about 70 loci that we had deeply sequenced. And on the y-axis is the cumulative frequency of all the alleles we found at these individual loci. And these color bars are basically telling you, the dark blue is telling you that the allele was exactly similar to OSB, okay, which is what we expect at some of these loci. But the green ones are the ones that are the most dominant loci, but they don't look like OSB. So in fact, what any particular genome almost looks like is a random mixture of all of these loci. And this is only looking at 100 loci. So you can imagine that it's even more exacerbated when you look at the whole genomes of these organisms. So the picture that's emerging and that we're trying to understand a lot more carefully, although we don't have the kind of deep sequences we'd love to have, is that you have highly diverse genomes living in these microbial mats. And what is the real million-dollar question is, what's the impact of having so many diverse loci, all essentially having the same function, so sort of like a pan-genome, but still with a lot of variation? What's the impact of that for evolution, for various other things that we're interested in? How do they deal with environmental stress? So for the last part of this talk, then, I want to move from diversity to what was in my title, the idea of conflict. So life is not just about getting used to environmental stress, for example, high light or nutrient stress. Often these guys have to deal with phages. And we know almost nothing about phages in the natural environment. And that's something, again, that I want to put out here, is that it's I think, very much the next frontier in terms of understanding how much diversity is there. And we got alerted to looking at this because of the CRISPR-Cas immunity system, which, by the way, when we discovered this, didn't actually exist, but in terms of knowing about it. But we found, in fact, that in our genomes, there were many, many of these Cas-CRISPR loci. And just to give you a 1-minute explanation of why these CRISPR loci are so interesting, there are ways by which a bacterial cell, if it's attacked by a phage or, for example, a plasmid, can actually take little parts of their DNA and acquire it into its DNA. It then makes these long CRISPR RNAs, which are then cut up into small CRISPR RNAs, and it's as if it's an arsenal waiting for the next attack. So if the cell is then attacked by a phage that is identical, at least in parts of its sequence, to the CRISPR RNAs, these CRISPR RNAs, through a very complex protein mechanism, can then basically chop up the incoming phage or plasmid. So it's what's been called an adaptive immunity system, and it's an extremely efficient way to get rid of phage, particularly phage that are already in the environment. Now, I know that most of you would probably think, and as Jean referred to, CRISPR loci as a method for gene editing. We actually think of it as a completely different mechanism, really, and that is as a way to understand coevolution. Now, how do I mean by that? I should preface this by saying almost all cyanobacteria and practically every archaea that's been sequenced so far have loads of these Cas CRISPR loci. And what's fascinating to me about them is that the way these spacers are acquired, they come in directionally. So they come in at the 5' end, and they tend to be lost at the 3' end. So it's almost as if it's a time-lapse image of which phages have attacked and which phages have left. So it becomes, in my mind, a very powerful way, given the fact that we have limited sequencing ability and knowledge about these, to watch the co-evolution of a host and its phage. And we're trying to do that. We're in the process of doing that. And so here's just a running list, just to give you a flavor of how we're doing it. So for example, OSA has 89 of these spacers. So think of these as little bits of phage DNA. OSB has 116. Here's a couple of other examples. These guys also happen to be in the mat. They're a big part of the mat. They're also phototrophs. These have a whopping 500 spacers. That's not an insignificant amount of foreign DNA to keep in your host DNA. And so what we're trying to do then is to use the fact that we have these host spacers from hosts that we can identify, and we use them almost like fishing hooks to pull out parts of phage DNA, because if a spacer is unique to OSB and matches any part of a phage DNA that we can get our hold on, it's a powerful way of putting these two pieces together. And why is that important? It's because we don't know very much about these phages, and we can't go back to any databases. There are no cyanophages so far, even sequenced from the hot springs. So that's what we're in the process of doing. But one thing we do know, which is exciting, is that if you take primers for viruses that parts of which have been sequenced, which is the work by Schoenfeld, he's at a company who's trying to find interesting new thermophilic polymerases. You can take parts of that virome and you can take our DNA from the mat, take these viral primers, and you get very significant amounts of PCR products, which is telling us that these viruses do exist and they're pretty populous in the mat. We can go a step further, and we're still in the process of trying to do this, where you do something a little more sophisticated, and that is that you can take a PCR primer that's upstream of the first set of of host spacer regions, and you place the second primer right in the repeat region. Now what you'll get, as you can see here, this is DNA from OSA, this is DNA from OSB. You get a ladder because you're getting a product from here, you're getting a second product from here, and these ladders exactly match what we expect them to be based on the genomes we have. And then you can get directly to the mat, and you also see a ladder, but it's much more messy, because you can imagine here now you're getting everybody who's there, not just a clean reference genome like OSA or OSB, but what it's telling us is that we can now start to recruit only the spacers from the mat DNA rather than just sequencing everybody who's there. So what we're interested now in is really exploring the dynamics of the host and the phage as a function both of time and the local environment. And we're in the process, although it's not trivial to get reference phage genomes, And I'd love to talk to people more about this. Part of it is that there's a lot of recombination going on even with the phages. So all the assemblers fail. You get these trees, and then you can't assemble the whole genome. We maybe need to sequence a lot more. We need to get a much larger database of CRISPR spacers. And then I think if we're able to use microfluidic devices to capture single cells, it'll sort of close the circle. We'll get the host population, all the spaces that are there, for example, at a particular time. But we'll also be able to see or get a profile of the spacers in one particular cell. And it may be that every cell or every cohort of cells really has a very different set of spacers, which to me is really fascinating. It's our first indication of the fact that the phages are making an impact on who's there and who's surviving. So in summary, what I've tried to tell you today is that because we don't work with E. coli or Vibrio or a human pathogen, we've had to build a lot of these tools ourselves, but I think we've made some progress. We have reference genomes. We've done a lot of comparative metagenomics. I told you some examples. We now have lab isolates, and using now tools of synthetic biology, et cetera, we're also trying to make these into more useful chassis for various things that we're interested in. And I hope I've sort of shown you at some level, not with a lot of detail, that the cyanobacteria seem to be extremely dynamic and diverse, both at a functional level as well as at a genomic level. And that's thanks to transposons, et cetera. And just a first glimpse of how important phages might be in these biofilms, something we really haven't considered in many environmental systems, although we know they're there. And the other thing I think that's overarching all of this is that these mapped biofilms, which we're very interested in, are developing and evolving as light-driven communities. And that's where the part that I'm also interested in is how is light playing a role in phototaxis, because these guys move up during the night. They move down into the mat when the light gets too high. So it's an extremely dynamic system. They're not just sitting in gel doing nothing. And a final thought really is that we can and do study model organisms in the lab, and we study them as individuals. But in fact, I think closing that circle of looking at them in the lab, but also generating hypotheses about what's happening in communities, by the way, some of the examples I've shown you, is to me probably the most powerful way to go at both ends of this question. So I'll just end by showing you some of the people. I just really want to focus on Michelle, who was a graduate student in my lab and is now continuing as a postdoc, and did a lot of the work on the virome and the metagenomics, really heroic work. And Mike Rosen, who we're working with on the microdiversity right now, and several other people who obviously along the way have helped with the work we've done for several years. So that's good on timing. So I'm happy to take questions. And if you don't have a question now, but wake up in the middle of the night thinking about cyanobacteria, that's my email. So thanks a lot.